Hi, everyone. It's great. Can everyone see my screen? Good. Very happy to be here. We are the Carnegie Mellon University team and University of Pittsburgh, and we're excited to be presenting for the Third Coast Augmented Intelligence for Health Bowl. Our design is going to be focusing on improving diversity, equity, and inclusion in clinical trials. I'm Alexander Chang. I'm joined by Kaylin Avanir, Aditya Singh, Anirudh Venkata Subramanian, Shishi Liang, Kate Guo, Samiksha Kale, and our advisor, Dr. Rima Padman.
Hi, everyone. Let's meet this encouraging lady here. This is Deborah Edwards. Deborah is a breast cancer survivor. Behind this encouraging smile, Deborah experienced cardiac side effects from breast cancer treatment, a problem more common in Black women. In the summer of 2017, Deborah began a treatment regimen known as AICTHP, which combines 3 chemotherapy drugs with 2 anti-HER2 medications. Soon, Deborah experienced severe side effects that almost killed her. Deborah was very fortunate that she had health providers who listened and were able to adapt her treatment. However, this does not hide the fact that she experienced significant trauma because the medication was never tested in a diverse clinical patient population. Not everyone could be as fortunate as Deborah. A recent special report found that the imbalance in clinical research inclusion leads to limitations in applying clinical data, therapeutic indexes, drug safety, and toxicity, resulting in poorer health outcomes for excluded populations.
How do we go about discovering the root cause of such a problem? Deborah is not alone in this fight. Less than 20% of FDA-approved drugs have clinical trial data regarding treatment benefits or side effects for Black patients. Moreover, while 43% of the US population is not white, less than 10% of the population in clinical trial design over the last 25 years were of people of color. Patients like Deborah are suffering because they were not included in the majority of clinical trial design, which showcase how big of an issue this is. Health disparity in clinical trial design is further compounded by the fact that people of color are disproportionately affected by diseases. For example, study has shown that there is a 38% increase in breast cancer mortality rate amongst Black women from 2000 to 2010. Having a more diverse clinical trial population can save lives, especially lives that are already disproportionately in diversity. Additionally, research has shown that $1.2 trillion could be saved if racial and health disparity gaps have been closed during the 2003 and 2006 period, meaning that increasing diversity in clinical trial design does not only save lives, but could also help decrease healthcare costs significantly.
And this is a growing problem, and it's receiving additional attention. But how can we actually address the fundamental causes of this disparity? Our solution stems from the Latin concept of justice, equality, conformity, symmetry, or fairness. It is the origin of the English word equity, AIquitas. We are AIquitas, an AI-based system to improve diversity in clinical trials by analyzing clinical trial design components using machine learning.
So what are the clinical trial components? What is the process to creating a clinical trial? The status quo of designing clinical trials are first coming up with an idea that comes from a physician or a team of investigators. The idea is then created into an initial protocol that includes sections such as background, rationale, objectives, and most importantly, inclusion and exclusion criteria, which is the main focus of our product. The next step is the initial protocol is sent to the provider's clinical research team for the first round of review to determine whether or not their study is feasible among the provider's patient population. The clinical research team then searches their EMR to find if there are patients eligible among their patient population. In order to approve it as feasible, there must be more patients, at least 5 times or greater than their target enrollment number, to account for the issue that may arise, such as patients choosing not to enroll. This process can go back and forth between the clinical research team and the investigative team, allowing the research team to make suggestions and the investigative team to modify their criteria until their target enrollment goal is met. Once the investigation is deemed feasible, the next step is submitting a final protocol that includes elements such as study design, final inclusion and exclusion criteria, procedures, and the schedule of these procedures. The final protocol must be then approved by the Food and Drug Administration and the provider's institutional review board in order to protect the patient's safety. They oversee and review the whole process as it continues. And once approved, the investigator can now receive a list of eligible patients and begin recruiting patients for their clinical trial. It is clear that the status quo of clinical trial design exists as a dyadic relationship between the clinical investigator and the healthcare providers, communicating on what is necessary to get the study done. But as you can see, there is a problem because the review process can be extensive and tedious for both sides, and sometimes the clinical investigators will drop their idea just because they are shy of their enrollment goal, which is what we don't want. This is where our product, AIquitas, comes in. Our goal is to change the status quo of the clinical trial design to take diversity into account while finding patients to enroll in clinical trials. So we are enrolling patients from different backgrounds, allowing representation from different races and ethnicities, such as patients like Deborah. Our research inspired us to think, how can we design a machine learning tool to analyze the inclusion and exclusion criteria that assist in clinical trial recruitment to improve diversity, inclusion, access, and opportunity to receive treatments?
So we realized early on that to really tackle the problem statement, we will need to consider the perspective of not only the clinicians designing trials, but also the potential recruits who could contribute to better clinical outcomes, but are completely detached from the system. To carefully address our problem statement from all angles, we break down our approach into two solutions that synergistically combine to form our intelligent system. Our approach, the first solution is an AI algorithm that can parse the relevant language of clinical trials to not only identify the language that is exclusionary, but also inform the trial designers to alter their criteria and allow for a more racially representative study. The first solution can be broken down into three major processes, which are all guided by our NLP pipeline. In the first step, the trial designer will upload their criteria document to our system, which will be evaluated by a regression model to output a diversity score indicating the racial representation that it allows for. Our NLP engine will be using state-of-the-art transformer models to obtain contextualized embeddings of the free text, which will then be trained for a regression task using open source data made available by the NIH. And to adapt our language encoder model to the clinical domain, we are using the Unified Medical Language System, which has been shown to improve performance of general models for tasks in the biomedical domain. In the second step, we will map the output of our regression model back to the input criteria to identify what parts of the text contributed the most towards the predicted score. These texts will be flagged and shown to the researcher for potential modifications or removals. And in the third and final step, the clinical health researcher will be able to interact with our system, modify the flagged criteria, and assess its effect on the predicted racial representation. So looking at the example here, Alex, could you go back? If the inclusion criteria contains prior therapy with palbociclib, our model may identify that as a contributor towards the low score. Subsequently, the researcher may decide to alter or expand that criteria by adding ribociclib or any other CDK inhibitor and obtain a measure of how effective their modification is towards increasing representation. Our data will come from clinicaltrials.gov, which is the world's largest database of privately and publicly funded clinical studies around the world. And we will then pair that with the All of Us Research Hub, allowing us to experiment with real validated EHRs. And the second part of our solution really expands our system into a platform for creating access to clinical trial opportunity by performing effective matching between patient information and the trials that they are most suitable for. So our approach to effective patient and clinical trial matching is based on encoding the inclusion-exclusion criteria and the patient information into a comparable representation. So as the first step, we will convert the inclusion-exclusion criteria into EHR coding standards. And in the second step, we will encode the patient conditions and comorbidities into the corresponding EHR codes. We will then compare the given patient's embeddings to embeddings of all upcoming trials and obtain similarity scores that will indicate how eligible a patient is for that trial. These similarity scores, along with other important information such as the location and preferences, will be used to re-rank the trials and present to the user the most relevant trials that they are likely to be eligible for.
Thanks, Aditya. Therefore, through a systematic two-pronged approach, we build a model to cover all the different aspects of the problem. We begin with the discovery and defining of the problem to research, analyze, and synthesize the currently available data to build our models out. We then transition to development and delivery, where we refine our prototype through comprehensive on-ground testing in collaboration with clinical experts and patient advocates. This slide represents our timeline here. In our phase 1, we focus on the design and development of our prototype in order to perform the following: the extraction and sorting of the relevant data from our clinical trials, to design a fuzzy matching tool to map the criteria to relevant codes using UMLs. Finally, the development of the tool to map layman language to criteria language that can further be worked upon. We then integrate these models and consolidate them with the natural language processing and variable autoencoder models with cross-validation testing. Simultaneously, we will also work on the design of user interfaces for clinical trial investigators and patients. Finally, we field test our prototype through collaboration and engagement with clinicians, patient advocates alike. This also includes the refinement of our user experience and interfacing. We've had the opportunity to communicate with a lot of different stakeholders, a wide range of stakeholders across the clinical trial process, and we've heard really positive feedback about our tool that we're planning to design. Now let's see how a product delivery will impact a wide range of stakeholders in the clinical trial process. The trial investigator derives direct visibility on inclusivity from our tool and any flagged exclusionary language. This consistent feedback process allows them to add, modify, remove language as needed in order to improve patient representation. Therefore, this induces efficiencies in the protocol design. Healthcare providers access relevant information seamlessly in order to make relevant recommendations for their patients on the suitable clinical trials. Patients have improved motivation and trust in the system through active participation in clinical trials. The tool also enables easy access to clinical trial information, which was previously burdened with clinical jargon that patients were not able to understand and really complicated requirements for eligibility. The FDA is finally equipped with a method to allow accurate, critical evaluation of the language included in trial designs to incentivize the creation of diverse, inclusive patient pools that truly represent the population that the product will actually be applicable for.
Based on the Schaefer model, if we could fix 1% of the health diversity issues in the American healthcare system, it estimates that it would result in more than 40 billion in economic output gains for diabetes and 60 billion for heart disease populations. Now, not all of this is due to clinical trials, but it highlights the impact of worse treatment, earlier death, and more adverse events for disadvantaged populations that are at stake. Furthermore, when we talk about the ethics, which is the focus of this competition, there are 4 key points of the traditional Hippocratic Oath: non-maleficence, beneficence, autonomy, and justice. However, in the wake of George Floyd and the resulting protests, the University of Pittsburgh medical class decided to modify the original text to include social justice and anti-racism components. We hope that by developing this tool, we can not only increase the opportunity for individuals to be able to access these better treatments, to participate in this system, but also for these treatments to do more good and less harm as a result, so that stories like those of Deborah are less common. These are our references. I'd like to thank our advisor, Dr. Rima Padman, and as well as various community and medical clinical trial stakeholders that have helped give us feedback as we develop this proposal. Any questions?
Judges? Thank you, team. I can go first. Maybe I'm a little bit perplexed because in your key stakeholders and clinical trials, you don't mention the drug manufacturers. And I have like so many more follow-up questions to that, but are you thinking this is a tool only for investigator clinical trials, initiated clinical trials? Because as you can imagine, the biggest people that have to lose most with anything changing in the way that they're doing clinical trials are the drug manufacturers, right?
Mm-hmm.
And
yeah.
The one thing that is, I think, not spoken enough is the issue of like pharmacogenomics. And one of the biggest issues why some of these different races have been excluded in clinical trials is for the reason that the clinical trials will not work for them, right? Their genetic makeup is such that it will just not agree with the drug. So there is a reason that some of these populations have been excluded. so that the drug can be approved based on statistics that are skewed towards the preferred population. So after making this statement, I'm confused if this is a tool for just investigator-initiated clinical trials, and or if it's not, you have to rethink your whole model with what the drug industry has in mind. I don't know.
So I'd like to answer that question in two parts.
Yeah.
In regards to the first part about the involvement of pharmaceutical companies, we agree that they probably have a lot to lose about having diverse clinical trials. It increases costs. That's why there have been existing incentives not to put in the effort. But that's how we're hoping to operationalize via the FDA. Currently, the FDA requires all clinical trials, whether investigator-led, pharmaceutical company-led, whatever, to publish essentially a diverse recruitment plan. But there's no enforcement mechanism because the language behind these are so complicated that it is incredibly easy to essentially make it so that there are certain things that are exclusionary but not necessarily directly relevant, such as, for example, HIV status, liver function, pulmonary function. Many of these are driven by algorithms that include race as a factor, but close examination of these algorithms indicate that they might not be the most correct in excluding certain races from these clinical trials, but it's such a complex problem. This is where AI can start discovering those hidden features. In regards to the second question, which was about pharmacogenetics regarding to why certain races ought to be excluded from clinical trials. If that is, while that may be the case for certain drugs, we do think that certain things like, for example, certain chemotherapeutics, certain immunotherapeutics, if they are intended to treat African Americans, should be included in the clinical trial process in the first place, because even if the drug is approved on a very clean, perfect data set, it is likely the patients themselves will not represent that clean of an existing health record that will then lead to all of those negative effects that we've discussed.
Thank you.
How does this take into affect the hesitancy in minority populations in participating in clinical trials? Because that ends up being one of the biggest complaints by drug manufacturers and developers.
Yeah, that's a very good question. Thank you for asking that. The history of medical apartheid against African Americans in this country is extensive, from the theft of African American bodies for American medical schools to literal experimentation during the slave era. We've tried to push back against this, or at least guard against this risk, by reaching out to community leaders as well as physicians who are leaders in this space in order to try and address this problem from a multifocal perspective. So one, we're increasing opportunity by reducing those systemic barriers that exist in that exclusion criteria. Two, we're building a patient-facing UI so that they can access and access it more easily. And then thirdly, we have this advisory panel that we've composed of that include people who are, for example, have been involved in community research efforts looking at neighborhood health in the city of Pittsburgh and are involved at like the city council level, as well as clinical trial investigators, as well as surgical oncologists. So for example, Dr. Evans is heavily involved in the Gateway Medical Society, which is a group of African American and ally physicians designed to address these disparities. So we're hoping to, trust will take a long time, but we're hoping to at least start making that effort by ensuring that this doesn't diminish any efforts to rebuild that trust and in fact enhances it. Because it doesn't matter if this health system regains all of that trust if the health system continues to have these walls in place that prevent them from entering the trial in the first place.